Welcome to this talk on stable isotopes in ore geology. My name is Glenn Bark, and I'm a senior lecturer in ore geology at Luleå University of Technology. So the topic today will be stable isotopes and how we use those isotope signatures in ore geology. And I will use a case study familiar to myself, which will be the origin of the Fäboliden orogenic gold deposit. First, in this talk, I will address what an orogenic gold deposit actually is, some characteristics of it. We will talk about which isotopes were used in this study and what the conclusions could be made from this. And I will finish off with a couple of slides on how we can use stable isotopes in also mineral exploration. Then first, the name orogenic gold. In the past, there were lots of different names like mother lode type, Archean gold only type, greenstone gold, etc., etc. All these names actually as being associated with different deposits or the timing of mineralization, different host rocks, and metamorphic grade then. A certain commodity, these are gold only deposits, so they will be the only commodity of economic interest is gold, and there will be some silver but very little base metals, basically ppm values of base metals. So there were a bunch of different names, and in the mid-80s people tried to sort of bunch these names together, or these different deposit styles together, and Bruce Nesbitt called them in his paper from 86 mesothermal gold. So that became the funnel word, so to speak, for all these different types of gold. However, the word mesothermal was a bit unfortunate, since there is an old reference from Lindgren in the 1930s stating that mesothermal is used for deposits which are formed at fairly low crustal depths. And these deposits could actually be formed for at least down to 15 at least kilometers down in the crust. So in the late 90s, David Grooves and co-workers coined the word orogenic gold, which they thought then would better reflect the orogenic processes that shape and form these gold deposits. So that's where the name stems from, the orogenic gold name. So orogenic gold then is a deposit which is formed in metamorphic rocks. We are in a subduction zone environment in the accretionary wedge, so to speak. We are at mid to shallow crustal levels. Most deposits formed at around 5 to 15 kilometers depth. And we have a compressional structural stress sitting here. So the term orogenic actually reflects the accretionary and collisional orogenic settings for this deposit. Orogenic gold constitutes roughly 1/3 of the global gold production. So it's a fairly important type of gold deposit worldwide. In this diagram, they have bunched together orogenic gold with intrusion-related gold, but intrusion-related gold is a minor part of this. So the majority of it is orogenic gold. The number one producer here are placer type deposits. So this is a slide to show the distribution of the really, really large orogenic gold systems in the world. These are the so-called giant districts, which has more than 500 tons of gold in one single deposit or in a district of different deposits. And then there are a couple of ones which are extremely large, with the Muruntau being the absolute number one with over 5,000 tons of gold, which is the single largest deposit, orogenic gold deposit known on Earth. And these deposits are so-called supergiant deposits, which are deposits which has more than 2,500 tons of gold. So this is a schematic model of the formation of orogenic gold deposits, how they are formed in a very, very schematic way. So we will start with the number 1. We need a source rock, which has very low concentrations of gold, but still a source rock. And then we push this source rock, we deform it, we heat it up during metamorphism and deformation. So we generate a fluid, and this fluid needs to be focused somehow. And it will be focused through some sort of pathway, which is typically a shear zone in the crust. And finally, we need some sort of trap. It could be a chemical or a physical trap for this fluid, which would cause the gold that the fluid carries to be destabilized and to precipitate the gold through certain precipitation mechanisms. We will go into that a little bit further later on here. But we're in the subduction zone environment, and this fluid that is generated here, mainly through the breakdown of chlorite, will constitute water, carbon dioxide, methane, and a range of different metals, of which gold will be the important one here. So we will start with a source rock which has gold in the ppb values, lower ppb, and we will finish off at sort of an ore concentration in the ppm range. So we need concentration processes, geological processes that concentrate gold in this fluid. In this fluid, when the mineralization has precipitated in this, well, let's say chemical trap, then we will have hydrothermal alteration around this quartz vein, for instance. The proximal alteration will be the quartz vein, and then we have a faint or vague distal alteration zone, which progresses into the regional metamorphic mineral assemblage. And that borderline isn't very clear most of the times. And this alteration halo is typically in the centimeter to decimeter scale, but it can be, for certain very large deposits, up to kilometer scale. And gold is distributed both within, hosted by the actual quartz vein, but also in the immediate host rock surrounding the quartz vein. A typical metamorphic grade for an orogenic gold deposit will be green schist facies conditions. So the black dot in this image then represents the sort of global average, which is roughly 2 kilobars and 350 degrees centigrade conditions. So most of the deposits investigated and studied are in the green schist facies, but there are a fair amount of deposits also in higher metamorphic terrains, such as Fäboliden. And we see here that the Fäboliden deposit, which we will talk about today, sits at 4 kilobars and 550 degrees. So looking at the genetic aspects, trying to understand how these deposits are formed, one thing that we are interested in is finding out the source of the fluid and the metal, the fluid that carries the metal gold. And for this, we use different types of stable isotope analysis. The ones that are most commonly used are oxygen, hydrogen, and sulfur. And we sample minerals such as quartz, amphibole, biotite, which will represent the fluid composition here, and different sulfide minerals, which will represent the source of the metal, of the source of gold in that case. So when we have looked at the oxygen and hydrogen isotope signatures, we will compare those to typical signatures of magmatic fluids and metamorphic fluids to see pretty much where our data lands in this diagram here. So what we can see here is that sulfur isotopes, they will give us the source of the sulfur and by association gold, We typically associate gold with certain sulfide minerals such as arsenopyrite, and then the sulfur in the arsenopyrite is believed to have carried the gold in a hydrosulfide complex in the fluid. And we have, looking at the sulfur isotope signatures, we see that we have a large range for sedimentary sulfur, also a fairly large range for magmatic sulfur, and then at the top of this one, we have the range for orogenic gold deposits, which overlap both magmatic and sedimentary sulfur. And that will cause a problem for us working with isotopes in orogenic gold, and it will be evident a little bit further on in this talk. So now I have talked a little bit about certain characteristics of let's say an average orogenic gold deposit. So now we will use one of the case studies, and we will talk about the Fäboliden deposit in northern Sweden. And this is a deposit where we have done quite a lot of studies, and one of the studies was on stable isotopes, and we will talk you through that part of the project for this deposit. But first, I want to mention the importance of knowing actually what deposit type you're working on. Why is it important to know that this is actually an orogenic gold deposit? When we classify a deposit, we look at a large range of different characteristics, such as the age, the structural setting, the host rock, metamorphic grade, the structural complexity of things, the metal association, different PT conditions, pressure, temperature, and we also look at the metal sources. And the metal sources is where the stable isotopes come in. And characterizing or classifying a deposit here is important for greenfields exploration. If we can investigate one of these deposits and find out which geological processes are important in the formation of this mineralization, we can use that knowledge to find out why the gold in Fäboliden sits where it does, and why it doesn't sit 4 kilometers to the north or 2 kilometers to the east, for instance. There are geological processes then that control the position and the location of the mineralization. So by trying to understand those better, we can use that knowledge to find more gold, sort of mineral exploration in a nutshell. The Fäboliden gold deposit is situated in the so-called Gold Line district, which is an array of gold-only deposits. And this district is situated to the southwest of the perhaps better known Skellefteå VMS district. And this is in northern Sweden. The Fäboliden deposit, which we will talk about today, holds about 60 tons of gold, and it has been reported as a sort of a low-grade, large tonnage type of deposit with 55 million tons at 1 ppm gold. But there are richer sections within this ore, and the let's say present plans for extracting the gold is by going only for the richer parts, and then the resource model ends up at 11 million tons at 3 ppm gold. So this is a little bit about the geology of the deposit. It's situated in metagraywackies and also bordering to metavolcanic rocks, all these surrounded by granitoid rocks of the so-called Revsund granite in northern Sweden, in Västerbotten here. And on the left-hand side in the image, you can see like a red snake, which is the gold mineralization. And crossing that is a section which you can see on the right-hand side. So you will see that it's a fairly steep dipping ore formation here, or mineralization. It's the red in this, which is the quartz vein carrying also and hosting the gold. So it's dipping towards east, and it has a set of younger dolerites, barren dolerites, cross-cutting the whole formation here. The structural setting for this deposit is like this, that we start off at the left-hand side of this picture, and we have two vein sets, one fault fill vein set, which is the steeper one, and then we have an extensional vein set, which is almost horizontal or very flat lying. So starting off with that type of package, if we compress this whole package in an east-western direction, the sigma 1 will be from west to east. We will fold these extensional veins and we will get boudinage in the steeper fault fill vein. And the right-hand side of the picture is pretty much what it looks like in the Fäboliden deposit today. So the hydrothermal alteration around this deposit. What we can see in these images, in the lower right-hand corner, there are two white arrows. The top one of that is crossing a quartz vein, and surrounding this quartz vein, we have hydrothermal alteration in the form of diapside, calcic amphibole, like actinolite, tremolite, and hornblende, and we also have biotite and some plagioclase and potassium feldspar in this system. But what is evident here is that we have a very narrow alteration envelope around each quartz vein. In this case, it's only a couple of centimeters. The deposit mineralogy, we have a quartz dominant vein system with a low amount of sulfide minerals, typically below 2–3% of sulfides. The sulfides that we do have is arsenopyrite and pyrite, and then we have an arsenide löllingite as well. And the gold is then associated to the arsenopyrite and löllingite in most cases. Gold also occurs as free grains in the silicate matrix, but hosted or very closely associated with arsenopyrite and löllingite is the typical setting of gold in this deposit. So then trying to figure out the fluid and metal source in this deposit, we will use isotopes for this. And we cannot use gold, as gold only has one natural isotope, so we will not get a signature from that. But we will use the association, the assumption of the association that gold is correlated with arsenopyrite, and the sulfur in the arsenopyrite is coupled to gold in the way that we think then that the gold is carried as a hydrosulfide complex in the fluid. So we measure the sulfide isotopes in arsenopyrite, which is associated to the gold, and assume that the two elements, sulfur and gold, in the arsenopyrite have the same source. That is the assumption that we build this one on. So we have our sample, which is in the middle of the picture, top, and then we make two types of samples from this rock or grab sample. First one is cutting the sample and mounting it in an epoxy resin for use under laser ablation isotope analysis. The other way is to take the sample and crush it down, and then under a stereo microscope hand pick the minerals that we are interested in. For instance, quartz and biotite, and sitting and actually then hand picking these very fine-grained minerals. And that type of sample will be used for a more conventional combustion analysis of the isotope. So taking the sample to the laser ablation lab, we will put it under the laser, focus the laser on it, and we will dig, since this is a destructive method, we will dig with a laser into the sample, into the arsenopyrite, and by doing so, we will liberate the sulfur from the arsenopyrite. The sulfur will react with oxygen and form SO2 gas. And this is the gas that we want to sample. So we take the sample from the laser ablation chamber and put this through a trapping system of gas chromatography systems in a series setup. So what we are doing here, I will use the pointer here. We introduce the sample this way. It goes down to a container filled with liquid nitrogen, and the freezing point for SO2 is -75. For air, it's 215, which means that we will freeze the sample in this U-shaped glass tube. We will freeze the sample, but air can still pass through. So by opening and closing the valve up here, we can get rid of as much air as we can. And then after closing the valve, we will thaw this part of the sample again, which makes the SO2 release, and we can push the SO2 onwards here to the next trap. And as you can see in the upper picture here, we use 3 different traps to purify the sample more and more to get rid of as much air as possible. So when we have the sample ready from the trapping system, we will put it into the mass spectrometer, which is the lower left picture, this one. And from that, we will measure the sulfur isotope data that we will get from each sample. So the data that we will then got from this analysis is from arsenopyrite, shows a signature of 34 sulfur isotope between +3 to -1 per mil. And for pyrite, it's +0.5 down to -1.5. So what is evident here is that we are clustering the data around 0 per mil in a rather narrow range. And in the past, this type of clustering around 0 per mil has traditionally been recognized as the sulfur in these minerals have a magmatic source. That is the conclusions drawn sort of in the past from this. So then we want to compare the Fäboliden data, which is the white line in the middle of the picture here, with different types of source rock. So what we can see from the red here, these magmatic rocks, we see that we have an overlap, but we also see sedimentary rocks, which have a very wide range of sedimentary or sulfur isotopes up here. So what we actually can say from the sulfur isotope data that we got from the sulfides is that we overlap. It could be either magmatic or it could be a sedimentary source for the sulfur. So the sulfur isotope signature is non-conclusive. It's not a standalone conclusion that we can make from this. We need to back this up with other data. So then building further on this, now we have tried to pinpoint the source of the sulfur and the metal thereby. So now we will work with the source of the fluid, and we then go for quartz and biotite. And at the top right corner here, I have outlined one of these quartz veins over a drill core sample. And the quartz and the other gangue minerals here then obviously represent the ancient fluid that once crystallized here. So by measuring the oxygen in the quartz and the hydrogen from the biotite, we will get a signature for the oxygen-hydrogen isotopes in the fluid here. So we will be provided with a fluid signature from the oxygen and hydrogen isotopes. And then we want to plot our data over the magmatic fluids and the metamorphic fluids to see where our data land. And these two fields are from Taylor, 1974, and it basically means that what is a magmatic fluid is that it's something that is water exhaled from a magma during the cooling. Metamorphic fluid is a fluid that is in equilibrium with metamorphic rocks or formed by dehydration reactions during prograde metamorphic conditions. And we will get back to this later on. So we do the analysis for oxygen and hydrogen isotopes, and we see that we end up at positive values for oxygen, plus 11 to roughly plus 13 per mil. For hydrogen, we land at minus 120 to minus 67 per mil. But these are mineral data, and to be able to compare fluid data, we need to recalculate the minerals into fluid data. And for doing this, we need to know what mineral we worked with, and we need to know what temperature it has crystallized at. Knowing what mineral, we totally assume that we know that this is quartz and biotite. And the temperature for this system, we need some input data from another study. So in the past, we did fluid inclusion work on this deposit, looking at the fluid chemistry or composition. And in that study, we found graphite associated to the arsenopyrite. And this graphite was possible to measure by Raman spectrometry and to get a temperature reading from the graphite, the crystal structure of the graphite, between 500 and 550 degrees centigrade. So we will use that temperature to calculate the fluid values from the mineral isotope data. So knowing our mineral, we're working with quartz, for instance, here, and we're at over 500 degrees centigrade here. So we will use, for instance, the Matsuhisa reference from 1979, calculating our data into fluid data. So the data that we get will land at plus 8.5 to plus 11.3 per mil. That is the oxygen data calculated from mineral to fluid. Then we will compare these data. In orange here is the Fäboliden data, and we will compare these with other waters, which are bracketed in red on the left-hand side here. So we have seawater, meteoric water, magmatic water, and metamorphic water. And what we're going to see here that we have a very clear, obvious overlap with metamorphic water, but there is also a very minor overlap with magmatic water. And this will cause a problem for us later on. So we're doing the same thing with hydrogen isotopes. We calculate this into fluid data, which then will land minus 85 to minus 32 per mil, and compare that with other types of water. And we see that the Fäboliden range in orange here, it will overlap with everything that we have except seawater in this graph. So a significant overlap both between magmatic water, meteoric, and metamorphic water. So we plot our data and compare this to the magmatic fluid, typical magmatic fluids, and typical metamorphic fluids. So the Fäboliden data is in gray shaded area here, with the black dot being the median of the oxygen and hydrogen isotope values. And we see, as one many times do in isotope studies, that we overlap both the metamorphic fluid and the magmatic fluid, even though the metamorphic overlap is stronger. So for orogenic gold deposits formed by fluid metamorphic fluids, the running hypothesis is that the fluid originates from dehydration reactions during prograde metamorphism, during breakdown of chlorite, generating the fluid, and the dehydration of hydrated minerals such as amphibole. We heat amphibole up, we will get pyroxene, and we will get water or a fluid. And this fluid will scavenge metals from the rock and transport them, and later on, hopefully then focused into some sort of pathway like a shear zone and trapped, precipitated in a mineralization. So since this is the running idea, we assume that we're dealing with a metamorphic fluid. But we have a small magmatic component in our data that we need to investigate further. So are we looking at a mixture between metamorphic fluid and a magmatic fluid, or how do we actually interpret these data? So before 2008, the conclusions from this type of study would be that the fluid originates from a magmatic and or deep-seated metamorphic source, meaning that we don't really know if it's magmatic or if it's metamorphic. We sort of bracket everything under one and the same stuff. So we see then from the oxygen and hydrogen data that we have this overlap. We see that from the sulfur data that it typically or historically was considered a magmatic source for them. So then the conclusion would be that, okay, this fluid originates from a magmatic source rock. But we still need to explain the little bit high oxygen 18 signature that we get for it, the so-called the magmatic source. So what then could cause an enrichment in the oxygen 18 isotope? Well, we can have sedimentary rocks in the reservoirs, as we can see here on the left-hand side, these types of rocks having a high oxygen value. Having these in the reservoir will push the oxygen isotope data up. So that would be one possibility. Another possibility is assimilation of country rock. If you have a magmatic intrusion, in assimilating sedimentary rocks into it, for instance, that will also generate a higher oxygen isotope signature for this one. And for both, there are, in the Fäboliden area, there are two main granitic suites, the Revsund and Skellefteå, and both these suites show signs of assimilation of sedimentary rocks in them from the Wilson study from the mid-1980s. So knowing this, we will then refine our conclusion or our model and say that, okay, we believe it's a magmatic source rock, but it has been crustally contaminated, basically saying that there are sedimentary rocks incorporated into the magmatic rocks. So we believe that is the origin of our fluid. But after 2008, that year, Chang and co-workers showed that the sulfur isotope range could be very much narrowed if subjected to metamorphism and strong deformation. So the very wide range of sedimentary isotope signatures that we see in sedimentary rocks can actually be pushed a little bit together. So we are sort of approaching magmatic sulfur signature in the data, in the sulfur data. Then in the Fäboliden area, we know that the deposit is mainly hosted by a massive sedimentary package. These are greywackies, which are probably more than 10 kilometers thick. So it's a very thick sedimentary pile. So then we feel that we can have then sulfur from the sedimentary rocks as well, which would fit also this signature, not just relying on sulfur isotope signature around zero being magmatic. It can actually also then be sedimentary in nature. So then talking about sedimentary sulfur, this is an SEM image, scanning electron microscopy image of an ocean floor sediment sample that we dried, and we looked in the microscope and we tried to find pyrite in this particular study. And these are the fine sort of nodules with the right arrow, the red arrow, sorry. And they look a little bit more like this, coming in close into detail. So this is framboidal pyrite, small aggregates of very, very extremely fine-grained pyrite formed at the ocean floor. And in 2009, Ross Lodge and co-workers showed that gold in pyrite formed in this way, in sedimentary pyrite, that these pyrite incorporate gold and other metals in them during precipitation. Gold is precipitated from the oceanic water into the pyrite crystal structure. And so we know then that we have a lot of pyrite in these sedimentary rocks. And during metamorphism, gold is released from the pyrite. When the pyrite passes over the green schist to amphibolite phase transition, pyrite will transition into pyrrhotite and sulfur will be released. And this sulfur would then help to carry the gold in the fluid that is generated from that. So the isotope data that we have in this study as far lets us basically argue that we have a metamorphic fluid, but we have a small magmatic component to this. We have a lot of sedimentary sulfur and gold here. So putting it all together, the orogenic model for Fäboliden would look something like this. This is a typical model for an orogenic gold deposit, how it's formed, one which is hosted in sedimentary rock. So we're starting off at the sedimentary source rock. We have gold locked in pyrite here in this area, the number 1 area. And then we push on this source rock at number 2 here. So we deform it, metamorphose it, and we will generate a breakdown of chlorite and pyrite. The chlorite will generate a lot of fluid. The pyrite will transform into pyrrhotite, and in that reaction, gold is liberated and carried by the fluid as a hydrosulfide complex. So at number 3, we have pushed this source rock. We generate the fluid, and the fluid is transported and focused then into, let's say, a shear zone would be a good thing. And then we need a precipitation mechanism. We need to destabilize this gold-carrying fluid somehow, and this could be done by that this fluid comes into a more iron-rich rock. So the sulfur will react with the iron, forming sulfide minerals, and by that being destabilizing the fluid and precipitating the gold as a mineralization and then hopefully as an ore. And in this case, there will also be precipitation of pyrrhotite, arsenopyrite, and gold together in this case. So this is a typical model for an orogenic gold deposit, how it's formed. Looking at the picture from Simpson, 1988, a little bit modified, we see that we have the same package. This is part of the accretionary wedge. We push this, so we have a metamorphism and generating this metamorphic fluid down here. It's migrating into this shear zone structure like this, letting the fluid migrate up the shear zone, and then for different precipitation mechanisms, perhaps coming into a more iron-rich rocks, or from repetitious pressure drops in this shear zone, actually seismic events, earthquakes, destabilizing the fluid and precipitating the gold. The yellow star here is where we then think that the Fäboliden deposit was formed at roughly 12 kilometers depth in a situation like this. So then going from the, let's say, the general orogenic gold model to the Fäboliden deposit. So in this deposit, we need to consider the magmatic component. And so a precipitation mechanism here would be that we have the metamorphic fluid carrying the gold, and then we have an input of a magmatic fluid, and the mixing of those two fluids causes these two fluids to be destabilized and for gold and sulfides to precipitate at the Fäboliden mineralization. So the mixing of these two fluids seems to be important, and it's corroborated by other studies that we have done on fluid inclusion analysis, also then indicating mixing to be an important feature in the precipitation of the gold at the Fäboliden deposit. So, and this is pretty much where the scientific front stands today when with concerns for the origin of the fluid source in orogenic gold deposit. There is a controversy. There are a bunch of scientists consider the metamorphic fluid to be the one and only, but and there is another camp saying that magmatic fluids needs to be considered as well. Sometimes we will probably have a mix of things. And in different deposits, the origin or the fluid source likely has different origins. So we figured out the origin of the sulfur and by that the gold and also the fluid at the Fäboliden deposit. But why do we do this now? Is it only for academic use? I use this to publish my data and I'm happy about that. Or can we actually use this in some other way? So what I would argue, and I would say that the mining industry is typically less interested in working with isotope geochemistry in mineral exploration. But I would argue that we understand the geological processes that are responsible for the mineralization at these different deposits in a better way working with isotope data. And it didn't work out in this case, but it would be a neat thing if we could actually pinpoint the signature for the fluid and pinpointing the isotope signature for oxygen and hydrogen for a certain rock type or lithology in the area that we're working. So let's say that in the map here, let's say that the intrusion A is related to the mineralization at Fäboliden and granite or intrusion B is not. Saying that the A intrusions are more interesting, we will definitely narrow the exploration targets here, and we can sort of focus the efforts in the mineral exploration and then hopefully find more gold. Another thing that we can do with isotope work is look at something called vectoring, which is actually a way of trying to see the mineralization from far away. So the number one here, then, this is a schematic image of a so-called blind ore deposit. The blind deposit means that it doesn't crop out at surface. We don't see it at the surface. So this little guy with a hammer doesn't see the mineralization down below here. But in the area, number one here, we have the hydrothermal alteration. We see the mineralogy, we see the arsenopyrite, we see gold, we see pyrite, but going a little bit broader. So halo number two then, we will not see the arsenopyrite, but we will see changes in the chemistry of certain minerals. We can see it in for instance, in biotite or pyrite, which is in these sedimentary rocks then. And also perhaps in whole rock data, but more likely in mineral chemistry data. And then going even wider, we will see changes in isotope signatures, which then is the number 3 halo. So out here, we might see differences in the oxygen isotope for, let's say, quartz. So then by taking a sample at the surface, investigating the oxygen isotope signature for quartz there, we can hopefully say that we have a good indication that there is something of interest below. So we are using the isotope changes in different minerals to see towards depth in a chemical way, not using geophysics, but using mineral chemistry in this case. So with those hopefully entertaining words, I want to thank you for listening. Take care and goodbye.